Good afternoon, everybody. Today I will present you a short summary of a project we are developing in the laboratory of Alcalá-Vida, focused on the study of striatal procedural memory-induced transcriptome and epigenome in Huntington's disease mice. As you know, Huntington's disease pathology is accompanied by a plethora of molecular mechanisms. And among them, our focus of research in the last years has been to better understand the mechanisms underlying transcriptional dysregulation in the disease. In this sense, we have recently described how early remodeling of chromatin structure in HD mice predominantly affects transcriptionally dysregulated genes, suggesting a possible causal interconnection between them. Consistently with the key role of these two mechanisms in memory formation, it still exists a missing gap between the role of these alterations and phenotypical consequences, such as cognitive alterations in HD. To answer that, we have made use of the transgenic R61 HD mice model and designed an interdisciplinary approach combining an in-house developed memory paradigm, the EHDN maze, with several biochemical, transcriptomic, and epigenetic approaches. For today's presentation, I will focus on the RNA-seq transcriptomic data and 4C-seq and ChIP-seq epigenetic data. As I have commented you before, the EHDN maze is an in-house developed task that Caroline Lutz, a former PhD student from the lab, adapted to mice from a previous protocol performed in rats. In this aquatic test, mice are released from a fixed arm and trained to find a hidden platform to escape from the maze, for which they can use spatial cues located around the pool, relying more on the hippocampus, or they can progressively automatize the task to locate the platform by turning right and then left, relying more on the striatal. Finally, a probe test can be done shifting the mice to the adjacent left arm and evaluating the first arm visit, providing information of whether the mice rely in a spatial strategy, more hippocampal dependent, or a procedural strategy, more striatal dependent. What we found when testing R61 mice was first an early deficit in the learning of the task, as shown by the reduced percentage of correct response, but also reduced preference for the procedural arm as the first arm visit, suggesting an impairment in striatal-dependent procedural memory. We then evaluated transcriptional programs induced by the EHDN maze in both wild-type and R61 mice by generating RNA-seq data at basal, early, and late stages of the EHDN task. We observed that EHDN-induced transcriptional programs were severely abolished in the R61 mice striatal. In addition, we characterized the physiological transcriptional dynamics induced by the task in wild-type animals. We showed an early induction of genes involved in transcriptional and epigenetic regulation, such as EGR1, as well as in MAP kinase signaling, which will induce a late set of genes related to cellular plasticity and cell-cell communication processes, such as collagen-related genes. Interestingly, both early and late EHDN-induced transcriptional programs were impaired in R61 mouse striatal. Trying to understand the mechanisms that could explain the deficient induction of early transcriptional programs, we investigated if these genes were modifying their chromatin spatial interactions with distal regulatory regions, a key process in their dynamic transcriptional regulation, in response to the EHDN performance, and if this process was impaired in the R61 mice. To do so, we used circular chromatin conformation capture, 4C-seq, a sequencing technique that evaluates the spatial interaction of particular gene promoters with their distal regulatory regions. And we focus on a set of immediate early genes or non-transcriptional induced control genes. As shown in these Chorosky graphs, overlap of promoter interactions between the data generated in basal or EHDN conditions, as depicted by the areas with stronger tonalities, was reduced at immediate early genes as compared to control genes, and suggested, as expected, higher activity-dependent chromatin spatial remodeling at immediate early genes. Interestingly enough, the extent of these changes was smaller in R61 mice, suggesting that this mechanism is altering the disease. We wonder then if late epigenetic mechanisms associated to memory consolidation could be equally impaired. And to do so, we generated chromatin immunoprecipitation coupled to sequencing, ChIP-seq, data focusing in two histone acetylation marks, H3K27 acetylation and H3K9 acetylation, associated with transcriptionally active chromatin regions and previously implicated in memory processes. We observed a selective modulation of H3K9 acetylation, but not K27 acetylation in response to the EHDN performance. And similar to our transcriptional data, this process was strongly abolished in R61 mice. When assessing the functional enrichment of these genes associated to a gain of H3K9 acetylation, we observed that they were implicated mostly in myelination and cell-cell communication processes, so more related to late consolidation processes of memory formation. So, as a brief summary, we have observed that in physiological conditions, the HH task is accompanied by the induction of different transcriptional waves, and that expression of immediate early genes responds to a higher dynamic chromatin spatial interactions with distant regulatory regions. In addition, late response genes appear to participate in long-lasting systemic memory consolidation processes and are reinforced by epigenetic chromatin remodeling of specific histone acetylation landscapes. In HD mice, in contrast, deficient learning and memory performance in the HH maze is accompanied by an induction of required transcriptional programs, as well as lower learning-induced spatial reorganization of immediate early genes. As a consequence, transcriptional signatures associated to memory systemic consolidation were totally absent, and plasticity-induced epigenetic histone acetylation was significantly diminished. So to finish, I would like to thank to everybody that participated in this project, especially to Caroline Lotz, who equally contributed to this work, and my supervisor, Karim Marian, as well as to my colleagues in Team 1 of the LNCA, to our collaborators and funding contributors, to the organizing committee for allowing me presenting you today this data, and to you for your attention. Thank you.